All right, let's jump right in. Today, we're going to break down one of the most incredible scientific feats ever, reading the entire human genome. We're talking about the literal blueprint for what makes you, well, you. So, how in the world did we actually pull that off? Let's get into it.
3 billion. Just let that number sink in for a second. That's our starting point. That's how many base pairs, or letters, are in the genetic code of a single human cell. It is a truly staggering amount of information packed into a space you can't even see.
So, to put 3 billion into perspective, imagine this. If you were to print out all those letters, you'd be looking at a stack of almost 3,000 textbooks. Now picture trying to find just one single typo somewhere in that massive library. That was the scale of the challenge that scientists were facing.
Which brings us right to the big question, doesn't it? How did scientists go from a microscopic bundle of DNA to a complete letter-by-letter sequence of our entire genetic code? It's a story that's all about brilliant tools and some incredible ingenuity.
This monumental task was taken on by the Human Genome Project, which kicked off back in 1990. It was this massive global effort to sequence and map every single one of our genes. But before they could read the book, they had to figure out how to even handle the pages.
And here was the first huge hurdle. DNA molecules are just enormous. I mean, even our smallest chromosome has almost 50 million base pairs. You can't just stick that under a microscope and start reading from one end to the other. So scientists knew they needed a way to break this impossibly long string into smaller, more manageable pieces.
And the solution? It actually came from nature itself. Scientists figured out they could build a kind of genetic toolkit using enzymes that bacteria have been using for millions of years. These became the molecular scissors and sorters that made this whole project possible.
So first up in the toolkit, the molecular scissors. These are called restriction enzymes. And the amazing thing about them is that each one is incredibly specific. It's like a key that only fits one single lock. It searches all along the DNA for a particular sequence of letters, and when it finds it, snip! It cuts the DNA right there.
This two-step process was really the crucial prep work. So first, scientists would use those restriction enzymes to chop the DNA into all these smaller, more manageable fragments. Then they had to sort them. To do that, they used a technique called gel electrophoresis. Basically, they'd put the DNA in a gel and run an electric current through it. The DNA fragments, which are negatively charged, start moving. And here's the key. The smaller pieces travel way faster and farther, which neatly sorts all the fragments by size.
So, okay, the DNA is now cut up and it's sorted by size, but that just gets all the puzzle pieces out on the table. How did scientists actually read the sequence of letters, the A's, T's, C's, and G's on each one of those tiny fragments?
This is where some really clever chemistry comes in. Scientists would make copies of the DNA fragments, but they added special dye-labeled bases into the mix. These special bases were basically stop signs. Whenever one of them was added to a growing DNA copy, the copying process would just halt right there. This created a whole collection of color-coded fragments, each ending at a different letter. Then, when you sort those by size, the sequence of the colors reveals the exact DNA sequence. Absolutely brilliant.
Okay, so they could read the small fragments. But how do you assemble thousands and thousands of them back into a complete chromosome? This is where shotgun sequencing comes in. The idea was scientists sequenced millions of random overlapping fragments and then fed all that data into a powerful computer. The computer then acted like the ultimate puzzle solver, just finding all the overlaps and stitching the entire sequence back together.
The progress since those early days has been just incredible. The original project took over a decade and cost billions of dollars. Today, we've got new technologies that can read a DNA strand directly as it's pulled through a microscopic pore, kind of like a thread through the eye of a needle. This lets us sequence an entire human genome in just a day or two for under $1,000.
So, after all that work, after finally reading the entire book of life, what did we find? Well, the results were definitely not what scientists expected. The genome was just full of surprises.
For starters, let's just take the number of genes. We humans, with all our complexity, we've got about 20,000 genes. That seems like a lot, right? Until you find out that a chicken has about 17,000 and a grape plant has over 30,000.
Yeah, this discovery led one research team to make this humbling and now pretty famous observation. It really drove home the point that an organism's complexity isn't just about the number of genes it has.
And this leads to one of the biggest surprises of all. Most of our DNA isn't even genes. Only about 2% of our entire genome actually codes for proteins. A full 50% is just made of repetitive sequences, and the rest is involved in regulating when and how our genes are used. So our genome is less like a list of ingredients and much more like a complex instruction manual.
Now, this massive scientific project isn't just some abstract thing. The ability to read our genomes has some really profound and personal implications for every single one of us. On the one hand, any two people are 99.9% genetically identical. But that tiny 0.1% difference is incredibly important. It means that about 1 in every 1,200 letters of your genetic code will be different from the person next to you. And all those tiny differences are what make you unique.
These little single-letter differences are called single nucleotide polymorphisms, or SNPs for short. Most people just call them SNPs. You can think of them as tiny variations that, when you add them all up, account for all the incredible diversity we see in the human population. Identifying these SNPs is what unlocks the power of personal genomics. It allows us to do some truly amazing things. We can trace ethnic ancestry, identify our personal risks for certain diseases, tailor medical treatments specifically to our unique genetic code, and even solve historical mysteries and help with criminal justice.
But of course, this incredible power comes with some really serious questions. Who should have access to this deeply personal information? Back in 2008, the US passed a law called GINA to help protect people from having insurance companies or employers discriminate based on their genes, but the ethical debate is still very much ongoing.
Which leads us with this final thought. The book of your life has been written, and for the first time in history, we have the ability to read it. But the question of who gets to hold the library card and what they can do with that knowledge, well, that's a story that we are all writing together right now.